All right, let's dive right in. We're going to talk about one of the most incredible, most essential things happening inside your body, literally right this second: DNA replication. And I promise you, this is not going to be like your boring old textbook. We're going to treat it like what it really is: a fascinating story about molecular machines.
I mean, really think about that for a moment. You started as just one single cell, right? And now you're made of trillions of them. How in the world does every single one of those cells get a perfect, identical copy of your original genetic blueprint? Well, that's the magic we're about to unpack.
So why does this topic feel so hard sometimes? Here's the thing. You open up a biology book and you just get hit with this wall of vocabulary. It's just a list of facts. But our brains? They're not really wired to remember lists. They're wired for stories. So that's exactly what we're going to do. We're going to turn this whole process from a list of facts into a story that actually makes sense.
Okay, so here's our game plan. First, we're going to look at that textbook problem. Then, we'll reimagine the whole thing as a cool molecular factory story. After that, we'll use a two-lane highway analogy to tackle the trickiest part. We'll lock it all in with a super helpful enzyme cheat sheet. And then, I'll give you a simple plan to make sure you've got this down for good.
So let's kick things off with Section 1, the textbook problem. This is all about getting to the bottom of why this can feel less like science and more like trying to learn a completely new and, honestly, pretty confusing language.
Does this list give you flashbacks? Yeah, it's basically an alphabet soup of enzymes, right? You've got Helicase, Primase, not one but two different DNA polymerases. It's so easy for all their jobs to get jumbled up in your head. Looking at a list like this, it's no wonder it feels a little overwhelming.
But what if we just looked at it in a completely different way? Now, this is crucial. Instead of a random list of words, I want you to start thinking of DNA replication as a super organized factory. Every single one of those enzymes isn't just a name, it's a worker. And each worker has a very specific, very important job to do on an assembly line. So let's jump into that new way of thinking. Section 2, a molecular factory story. I want you to forget about complex chemistry for a minute. Just picture DNA as a simple zipper. And we're about to go meet the crew at the factory that's in charge of copying that zipper.
And when you look at it this way, it all becomes so logical. First up, you've got the unzipper. That's Helicase. It comes in and opens the whole thing up. Simple. But you don't want it zipping right back up, so these little clips, our SSB proteins, come in and hold the strands apart. Now, all that unzipping creates a lot of tension further down the line, so the stress manager, topoisomerase, shows up to relieve it. The main builder can't just start working on a bare track, so Primase comes along and lays down these little start here signs. And only then, once everything is prepped, does the main builder, DNA Polymerase 3, come in to actually build the new strand. You see that? It's a logical sequence. It's a story.
Okay, deep breath. Now we're going to tackle the part that trips almost everyone up. Section 3, the two-lane highway. This is where we finally solve that confusing 5' to 3' puzzle. It sounds really complicated, I know, but trust me, with the right picture in your head, it's perfectly clear.
So here's the master rule. This is the key to everything. Each strand of DNA is like a one-way street. It has a direction. We call those directions 5' and 3'. And our main builder, that DNA polymerase, it's a little particular. It can only build in one direction, from 5' to 3'. Think of it like a road paver that can only move forward, never backward. And this one little rule creates this really fascinating problem that the cell has to solve.
And this slide shows the brilliant solution. Because the two DNA strands run in opposite directions, the cell has to use two totally different strategies. One new strand becomes the easy highway, we call it the leading strand. On this side, the builder can just move right along with the unzipping fork, building one long, continuous piece. Easy. But the other side, that's the complicated highway, or the lagging strand. Here, the builder has to move away from the unzipping fork, which means it has to build backwards in a bunch of short, separate little pieces. And that requires a whole cleanup.
Let me break it down for you. So on that complicated highway, the cell gets really clever. First, Primase puts down a start here sign. Then DNA Polymerase 3 jumps on and builds a short little piece backwards away from where the zipper's opening. Then, as the zipper opens up more, the process just repeats. Another primer, another little fragment. So now you've got all these separate pieces. That's when the cleanup crew arrives. A different enzyme, DNA Polymerase 1, comes in and removes all those starting primers. And finally, DNA Ligase acts like superglue, sealing all those little fragments together into one perfect, complete strand.
Whew, that was a lot of different workers, I know. So let's make sure we have them straight. We're moving on to Section 4, the enzyme cheat sheet. This is just a simple, powerful trick to make sure all those names and jobs get locked into your brain for good. You want to know the secret to remembering all this? It's a mnemonic, a silly sentence. Your brain loves this stuff. Just remember, happy students teach people different donuts lovingly. Each word cues an enzyme and its job. Happy is for H-helicase, which opens the helix. Students is for SSB proteins, which stabilize the strands. Teach is for Polymerase, which tackles tension. See how that works? Different donuts helps you remember the two polymerases. DNA Pol 3 does the building, while DNA Pol 1 deletes the primers. And lovingly is for Ligase, which links everything together. It's a little goofy, I know, but it really, really works.
All right, so you've got the story, you've got the memory tool, so what's next? Well, Section 5 is your mastery plan. Because it's one thing to watch this explainer, but it's another thing to really own this knowledge. Here's a simple 3-week plan to get you there. Look, please don't try to cram this the night before an exam. Just spread it out. Week 1 is all about understanding. Watch this, take your own notes, and then, this is the most important part, try to draw the whole replication fork from memory. In week 2, you apply what you know. Find some practice questions. And even better, try to explain it to a friend. If you can teach it, you know it. Then, by week 3, you're ready for mastery. Find some old exam questions and practice drawing it all out under a time limit. That's how you build knowledge that sticks.
And before we finish, let's just zoom out for a second and look at the big picture. This whole process has a name, semi-conservative replication. It sounds fancy, but it just means half conserved. Each of the 2 new DNA molecules you end up with has one of the original strands and one totally brand new strand. The cell conserves half of the original. This comes up on exams all the time, and now you know exactly what it means.
So let's do one last super quick recap. The entire story can be boiled down to 3 main steps. Unzip the DNA, stabilize the open strands, and then build the new ones. This process creates one smooth, continuous strand and one fragmented strand that gets stitched together later. And the final result? 2 perfect, identical copies of the original DNA molecule, all ready for a new cell.
And there you have it. You've just walked through this beautiful, complex dance that literally keeps life going. The cell's copy machine isn't some big, scary mystery anymore. So now that we've decoded this process together, what amazing biological story should we dive into next time?